Good morning, all of you. Now, today we are going to discuss enzymes. Now, see, to learn English, alphabets are important. The knowledge of alphabets is important to understand the language. Same is the case here. To understand biochemistry, the knowledge of enzymes is very, very important. Because how do we define this biochemistry as a subject? Simple, it is the chemistry of the biomolecules. What reaction is occurring in our body? And what are enzymes? Enzymes are the biocatalysts which are actually increasing the rate of that biochemical reaction. So whatever biochemical reaction is there, whether it has to undergo, it has to go, whether it has to be stopped for some time, everything is controlled by these enzymes. So today we are going to discuss part 1 of enzymes. First of all, to start with, you should all be familiar with this term enzyme. So enzymes are what? They are the biocatalysts which increases the rate of a reaction. In general, if I have to write a reaction, this is the reaction. Enzyme is going to combine with the substrate, which in chemistry we call as reactants, going to form a enzyme substrate complex which cannot be there for a longer time, and it will be dissociated to form the enzyme and the product. So this is a typical biochemical reaction we are talking. So what is the function of enzyme? Enzyme is going to increase the rate of a reaction. And how they are going to increase the rate of a reaction? By lowering down the activation energy. Now let's take a reaction. It's like this. Whether it is exothermic or whether it is endothermic, that depends upon the enthalpy of the reactants or the products. Now see, say these are the reactants or the substrates. They have to cross this energy barrier so that they can be converted to the product. This is the state of the formation of this enzyme substrate complex. Now you can see these reactants have to climb up to this much, then only they will be converted to products. What happens is this reaction is without the presence of the enzyme. If we add enzyme into this reaction will be like this. So you can see we have what? Decreased the time. If a reaction was occurring without an enzyme in say about 1 minute, in the presence of enzyme, that time will be decreased. And how? Because they are what? They are lowering the activation energy. Here you can see in the first one may the reactants have to climb this much to be converted to product. But in the second reaction, in the presence of the enzyme, it has to climb this much so that it can be converted to product. So enzymes are the biocatalyst. Again, I am telling you, they increase the rate of a biochemical reaction by lowering down the activation energy. And this is a simple biochemical reaction. Now, just now I told you that biochemistry is nothing without enzymes. So enzymes can be of so many types. Their occurrence can be different. We can say if an enzyme is present inside the cell, it will be a intracellular enzyme. If an enzyme is present outside the cell, it would be extracellular enzyme. If an enzyme has only one unit which is active, it will be monomeric enzyme. And if an enzyme has two active sites, it will be a dimeric enzyme. Similarly, on the other hand, you can say that there are certain multi-enzyme complexes also. Multi-enzyme complex means enzyme cannot work alone. It requires so many other things to work with it. So depending upon this, enzymes are broadly classified. Depending upon the occurrence, they are broadly classified. So now, see, we are going to take each aspect one by one. Overall, enzyme is nothing but a protein. So you can say all enzymes are proteins except one, which is ribozyme. Because here you can see ribozyme means RNA has a catalytic activity. So wherever RNA is functioning as a, you can say as a functional group, then it won't be a protein. The example of ribozymes are, see, peptidyl transferase. Peptidyl transferase, which is a very, very important enzyme of translation. Ribonuclease. So these are what? These are not proteins in structure because they have that RNA in them. Except these, all enzymes are proteins. Now, if an enzyme has to work, just now I told you, it cannot work alone. So it has a non-protein part also associated with it, which is known as the prosthetic group. Now, if this prosthetic group is a metal ion, very, very important. If it is a metal ion, for example, copper, zinc, magnesium, manganese, iron. There are other examples also, selenium. Then it will be known as a cofactor. Means if this protein part of an enzyme is associated with a non-protein part, which is a metal ion, it will be known as a cofactor. And if it is attached to a organic molecule, like B1, B2, B3, B5, B6, B7, these are all types of vitamin B complex, it is known as a coenzyme. So there is a difference between a cofactor and a coenzyme. Cofactor is when the protein is associated with a metal ion, and when this protein part is associated with the organic molecule, it is a coenzyme. Just the protein part of the enzyme is apoenzyme, but apoenzyme plus cofactor or coenzyme is known as the holoenzyme. Again, very, very important. So now by just seeing this, you can differentiate between a cofactor, a coenzyme, an apoenzyme and a holoenzyme. So this is how the enzyme is acting. And how enzyme is acting? Enzyme is acting on the substrate, forming this enzyme substrate complex to give enzyme and the product. Then next we come over to, here I'll give you some examples of this. They say you must be familiar with like copper. Copper is a cofactor for cytochrome oxidase. Iron is a cofactor for cytochrome oxidase as well as catalase and peroxidase. Similarly, selenium, a very important trace mineral, it is a cofactor for glutathione peroxidase. So these are some of the, you can say, examples of the cofactors. Similarly, if we see the coenzymes, coenzymes में PLP or B6 is a coenzyme for transamination. Biotin or B7 is a coenzyme for carboxylation. Similarly, PLP is also used for decarboxylation. TPP is used for oxidative decarboxylation plus transaldolase reaction, transketolase reaction. So this is what, these are some of the coenzymes and the examples of cofactors. Then another important segment is, what do you mean by the activity of the enzyme or enzyme activity? Now see, it is clear from the word itself how active an enzyme is. Suppose, say, a teacher is delivering a lecture in the class and 50 students are sitting. It's very rare that all the 50 students are active at that particular time. So if the student is active, automatically the activity of the teacher would be more. Because whatever the teacher is telling the students, students are going to grasp it evenly and they are going to give good results. And that is what is the activity of the enzyme. How active an enzyme is means how actively it is combining with the substrate to form maximum product. Or you can say it has different units. Just say IU per liter is there. Or you can say conversion of 1 mole of a substrate in a second will be the activity of the enzyme, how fast it is going to react with the substrate. This is the international unit, IU per liter. One more unit is there, kettle. And otherwise it is micromoles per second. So this is enzyme activity, and overall if the activity of the enzyme would be more, automatically turnover number of the enzyme would be maximum, which is known as Kcat, catalytic constant for an enzyme. For example, trypsin has the highest Kcat number or highest turnover number. So these are the two very, very important definitions. Now see, the reaction is actually based on the activity of the enzyme. So what can be the factors which can influence the activity of the enzyme, or factors which can influence that? So the factors affecting the enzyme activity, the most important factors are temperature, pH, and substrate concentration. These are the three very, very important factors which are influencing the activity of the enzyme. Now see, if we take pH, we know enzymes are catalyzing all the biochemical reactions which are taking place in the body. So we can say that activity of the enzyme would be maximum at temperature 35 to 40 degrees. This is known as optimum temperature, or this is known as temperature optima. And if we see, if we have to plot the graph between substrate concentration and the temperature, it comes out to be a bell-shaped graph. Here, the activity of the enzyme would be maximum, because here the temperature would be 35 to 40 degrees. So, the influence of temperature on the activity of the enzyme is, it has a specific temperature optima between 35 to 40 degrees. If we decrease the temperature, if we increase the temperature, enzyme will denature. Means, because denature means it will be, you can say, distorted from its original nature. Because just now I told you enzyme is nothing but a protein. And protein is nothing, it is formed of the amino acids. So obviously if we increase the temperature, we decrease the temperature, number of bonds will break. Then comes pH. Again, every enzyme has an optimum pH which is roughly between 5 to 8. Little bit towards acidic, little bit towards alkalinity. But There are certain exceptions to it, like alkaline phosphatase. It works between 9 to 10 pH. Pepsin works at very acidic pH, that is 2. But overall, if we see, all the enzymes will show a pH optima between 5 to 8. And again, a bell-shaped graph will be obtained like this. This will be the pH optima. Then we come over to the most important factor which is affecting, because ultimately it is the substrate which is there. So what is the effect of this substrate concentration on the activity of the enzyme? Now always remember, this substrate binds to the active site of the enzyme. Active site, or it is also known as the catalytic site. So what happens is, more the enzyme, active site of the enzyme, more will be the reaction of the substrate. But a stage will come when all the active sites of the enzyme will be occupied by the substrate. Say, suppose if I say an enzyme has 10 active sites. Obviously, it is going to accept only 10 substrate molecules. Once it has reacted with those 10 substrate molecules, there is no active site left in the enzyme to bind with more of substrate. So it is going to attain a saturation kinetics. Very, very important. Saturation kinetics, or it is also known as steady state kinetics. And this type of graph is a hyperbolic graph. So you can see there are basically 3 important factors which are influencing the activity of the enzyme: temperature, pH, and substrate concentration. If we see the effect of temperature and pH, the graph is bell-shaped and it has a specific temperature optima and it has a specific pH optima. Third important factor is substrate concentration. More the active site, more will be the enzyme catalyze reaction, but a stage will come when all the active site of the enzyme will be occupied by the substrate. Then it is going to attain this steady state kinetics or saturation kinetics, or it is also known as zero order kinetics, which I am going to take after this. So these are the 3 important factors. Other factors can be inhibitors are there, activators are there. They will automatically either increase the activity of the enzyme or they will decrease the activity of the enzyme. But these are the 3 important ones. Then we come over to another segment. Another segment of this is enzyme kinetics. Again, very, very important. Kinetics means, as the name suggests, how a reaction is occurring. Because if we look at this reaction, basically when a reaction is occurring, we have to concentrate on the amount of the product which is produced. If the amount of product produced is what is required, then only there will be some authenticity of the reaction. So this we cannot measure. This enzyme substrate complex, okay, whatever is, always remember, whatever is written in square brackets, it is very unstable. So either it will be converted here or it will be converted back. So if we have to define the rate constant here is, so K1 would be the rate of formation of ES, K minus 1 would be the rate of dissociation of ES back into enzyme and substrate. K2 will be the rate of formation of the product and K minus 2 would be the enzyme substrate complex which is not converted into the product. So this is how we are going to define this. And kinetics is what? There were actually 2 scientists, Michaelis and Menton. They gave 2 terms. One was V0 and one was Vmax. V0 means initial velocity, means means just the reaction is just started. And Vmax is the final velocity when the reaction is accomplished. So whatever is the gist means whatever amount of desired product is produced is what is the target of this enzyme catalyzed reaction. And this equation given by Michaelis-Menten was V naught is equal to Vmax S over Km plus S. Very, very important. Now, what is this? Vmax, this is maximum velocity. This Km is the Michaelis-Menten constant. This is the Michaelis-Menten constant, which is actually telling us about the entire rate of the reaction, that how much product will be produced. And if we see the graph also, just now I driven the graph, it's like this. You saw that when the concentration of the enzyme is more, substrate is more. But at this position, it attains a saturation kinetics. Michaelis-Menten called it as steady state kinetics and zero order kinetics also. Why? Because now the substrate and enzyme are independent of one another. That is why it is zero order. It has attained a steady state. That is why it is steady state kinetics, and saturation is there, so it is saturation kinetics. Now, a very important inference we draw from this equation, that when initial velocity is half the maximum velocity, when initial velocity is half the maximum velocity, this Km is equivalent to the substrate concentration. Very, very important again. Somewhere in the middle of the reaction, this substrate concentration will be equivalent to Michaelis-Menten constant. Somewhere here. So this is the equation of kinetics of enzyme. V naught is equal to Vmax S over Km plus S, which is for a single substrate or a mono substrate. When obviously we talk about bi-substrate reaction, the mechanism is altogether different. We call it as a ping pong mechanism. But Michaelis and Menten gave a equation for this mono substrate reaction. And this is how it goes. Now see, what happens is, this is what we are calculating Vmax, and we are calculating, obviously every enzyme will have its own Km. This is again very important. So what is the significance of this Michaelis and Menten equation? Very, very important. What is its significance? Significance is what? Because see, it is actually influencing the activity of the substrate with the enzyme. Significance is it defines the affinity of the enzyme with the substrate, and it is inversely proportional. Very important again. Inversely proportional means if an enzyme has high Km, again I am repeating it, if an enzyme has high Km, the affinity of the enzyme with the substrate will be low. And if the enzyme has low Km, the affinity of the enzyme with the substrate will be more. And that is what exactly happens when we discuss about the difference between glucokinase and hexokinase. Now see, glucokinase has high Km. High Km means it has a very lesser affinity with glucose. And all of us know glucokinase is present in the liver, and it is active only when the concentration of glucose exceeds 100 milligram per dl. On the other hand, hexokinase is the first enzyme of glycolysis. Very, very important enzyme, whatever amount of glucose we are eating, because glycolysis plays a very, very important role in RBCs. So hexokinase has low Km, means it has a very high affinity with glucose. Means at the basal concentration of glucose also. Means when the concentration of glucose is say 0.001 molar also, then also hexokinase is going to act. The reason behind this is because RBCs, whether they are in the well-fed state, whether they are in the starvation state, or they are in the fasting state, they have to depend on glucose for energy. So hexokinase plays a very important role there. So hexokinase has low Km, high affinity. On the other hand, glucokinase has high Km, low affinity. And the significance of Km is it is depicting the affinity of the enzyme with substrate. Then another important aspect of this Michaelis-Menten constant is Lineweaver-Burkes plot. What happens is, suppose we have to calculate the slope and the intercept. What we do is we reciprocate this equation. The Michaelis-Menten equation, if we reciprocate this, इसको उल्टा कर दो, तो it comes out to be 1 upon V naught Km Vmax S plus S over Km Vmax S over Vmax S. This cancels. And what we have to do is we have to plot a graph like this. The graph would be converted into an equation of straight line. And this will be the slope and this will be the intercept. Because the equation of straight line is equal to y is equal to mx plus c. This becomes the slope and this becomes the intercept. So what we have done is we have reciprocated, इसको उल्टा कर दिया, and this type of equation is known as Lineweaver-Burkes plot. Lineweaver-Burkes plot, or it is also known as double reciprocal plot. So basically what we are doing is we are calculating the slope and the intercept. So this is what is the kinetics of the enzyme explained by two scientists, Michaelis and Menten. Then we come over to another aspect of this. In fact, the most important aspect of this, the classification of enzymes. Very, very important. Now enzymes, if we see the earlier classification of enzymes, it was a very tedious one. Tedious one in the sense that depending upon the class, subclass, group, transferred moiety, it was classified. This is 1, 1, 2, 2. It was like this. 1, 1, 2, 2. Class, subclass, function, moiety transferred. So it was very difficult to memorize also. So according to International Union of Biochemical Nomenclature, enzymes are divided into 6 major classes. O, P, H, L, I, N. These are the 6 major classes of enzymes. O stands for oxidoreductases. T stands for transferases. H stands for hydrolases. L stands for ligases. I for isomerases. And again L for lyases. Now see, oxidoreductases enzyme catalyzing the oxidation as well as a reduction simultaneously. And that is what is actually acting in a biochemical reaction. If we see, all the biochemical reactions are typical redox reactions. Redox reactions in the sense, if something is being oxidized, the other has to be reduced. So oxidation and reduction takes place simultaneously, and all the enzymes catalyzing this will come under oxidoreductases. And this class is considered to be a very elaborative class because it has other subclasses also, like dehydrogenases, oxygenases, oxidases, and so on. Peroxidases, catalases, they will come under this. An example of oxidoreductases are LDH, PDH, xanthine oxidase, they will come under oxidoreductases class. Transferases, it is clear from the word itself, there is transfer of something. Now, whether there is transfer of phosphate kinase, whether it is transfer of amino group, it is aminotransferases. Example of kinases can be glucokinase, hexokinase, pyruvate kinase. Aminotransferases, the examples are alanine transaminase and aspartate transaminase. So there is transfer. Again, this is a very important question too. Hydrolases, all the digestive enzymes will come under this, like whether it is peptidases, lipases, nucleases, and so on. Ligases comprises of two classes. Either there is formation of something, synthetic, or synthesis is there. They say glutamine synthetase is there. And DNA ligase, which is also known as the glue of DNA, because it helps in sealing of the nicks which are caused in DNA. Isomerases, normal isomerization, you can say glucose 6-phosphate is converted into fructose 6-phosphate. Or other examples are, there are racemases will be there, epimerase will come under this, and isomerase will come under this. Then we come over to the last class, lyases. Lyases is splitting up class. For example, aldolase is there, which splits up. Fumarase is there. So we can say enzymes are broadly classified into 6 major classes: O, T, H, L, I, L. Oxidoreductases, transferases, hydrolases, ligases, isomerases, and lyases. Now, after studying the classification, next we come over to again a very, very important concept in this. Now see, if a reaction is occurring, because I told you, enzymes are catalyzing all the biochemical reactions in the body. So there has to be some mode of inhibition also. ऐसा तो नहीं है कि if the production starts, वो production होती रहेगी. There has to be a stoppage to something. So there is a type of inhibition which is occurring, which we call as enzyme inhibition. Very, very important mode. Now overall, if we see, we can classify inhibition. Different books have given different classification of inhibition. But here I am going to take all the inhibition separately. One is competitive inhibition. One is non-competitive inhibition. One is uncompetitive inhibition, and the most important inhibition is allosteric inhibition. Now, first of all, let's take this competitive inhibition in detail. Now, see, the name suggests you can very well make it out from the word. There is a competition. Competition in what? Enzyme, normal reaction. Enzyme is combining with this, forming enzyme and product. In case of competitive inhibition, there is a competition between the substrate and the inhibitor. Why? Because the inhibitor resembles substrate in structure. So what happens is, this enzyme, because the active site is there, this enzyme, instead of binding with the substrate, it can bind with the inhibitor. So when the enzyme inhibitor complex is there, no reaction will be there, right? Now what will be the kinetics of the enzyme in competitive inhibition? If we see, there are two factors influencing this. One is Km, other is Vmax. So let's take Vmax first. Now just now I told you there is a structural resemblance between the inhibitor and the substrate. So whether the inhibitor is binding, whether the substrate is binding, Vmax will be same because the reaction is going to occur at a normal pace. But Km will increase. Why Km increases? Because the affinity of the enzyme with the substrate decreases. Right. So in competitive inhibition, there is a competition. Km will increase, but Vmax will be same. Now, if we increase the concentration of the substrate, the reaction will be reversed. Or you can say the inhibitor will no longer be available to combine with the enzyme. That is why this competitive inhibition is also known as reversible inhibition. It can be reversed. Now, example of competitive inhibition are succinate and malonate. Succinate is a substrate for succinate dehydrogenase enzyme which converts succinic acid to fumaric acid. Now, if malonate is there, obviously there won't be any reaction. So, this is an example of competitive inhibition. Now, there are certain therapeutic use of competitive inhibition also. For example, allopurinol. Allopurinol is a competitive inhibitor of xanthine oxidase. Similarly, all the statins which are given actually for hypercholesterolemia, they are the inhibitors of HMG CoA reductase. So this is what? This is the therapeutic use. Similarly, all the anti-cancerous drugs. Anti-cancerous drugs are the competitive inhibitors of methotrexate. This is the drug and the enzyme is THF reductase. Right. So this is what is competitive inhibition. Then we come over to, I hope it is clear. So we can say that in competitive inhibition, Km will increase, but Vmax will remain the same. And competitive inhibition is also known as reversible inhibition because if we increase the concentration of the substrate, we can reverse this reaction. Then we come over to the second inhibition, which is non-competitive. Now see, non-competitive means no competition. As the name suggests, very simple. There is no competition between the substrate and the inhibitor. So what will happen is the inhibitor can bind with the free enzyme also, and inhibitor can bind here also. So if the enzyme is binding with the enzyme, if the inhibitor is binding with the enzyme substrate complex, it is going to form this. Now, whether it is binding here, whether it is binding there, automatically there will be change in Vmax, because rate of reaction will automatically decrease, because if it is binding with this enzyme substrate complex here. On the other hand, Km will remain the same. So in non-competitive inhibition, Vmax will be decreased and Km will be same. Now this non-competitive inhibition can be reversible, it can be irreversible. That depends upon the catalytic site of the enzyme where it is actually binding. Suppose it goes and binds to the somewhere near the active site of the enzyme. So again the functional unit of the enzyme will be affected. So it will be irreversible. And if it is binding somewhere else, not affecting, only Vmax will decrease, Km will remain the same. So non-competitive can be reversible, it can be irreversible too. Example of this is iodoacetate. Then third inhibition is uncompetitive. Uncompetitive means nothing is there. Very rare. And what happens here is Km and Vmax both are decreased and enzyme substrate complex binds only with the enzyme substrate complex. This inhibitor is going to bind only with the enzyme substrate complex. So you can see there is a difference between these three types of inhibition. In competitive inhibition, reversible inhibition, we can say Km increases whereas Vmax remains the same. In non-competitive inhibition, it can be reversible, it can be irreversible. Here, Km will decrease, whereas in non-competitive, Vmax will decrease and Km will be unchanged. But in uncompetitive inhibition, which is actually very rare, in this both Km and Vmax will be lowered. So, just now we discussed what? We discussed about the definition of the enzymes, how enzymes are reacting on a biochemical reactions. What do you mean by enzyme activity? What are the factors which are affecting the activity of the enzyme? Then obviously the classification, which is a very important part of enzymes. And last but not least, we discuss about the inhibition. Now in the next class, we are going to take a very important mode of inhibition, which is allosteric inhibition. So if you want to know the details about this allosteric inhibition, kindly subscribe to my channel and stay in tune with more of the biochemistry topics. Thanks a lot. Have a good day.